Let's begin with something that sounds like it belongs on a motivational poster in a dentist's waiting room. Stress is bad for you. You know this? Everyone knows this. It is perhaps the least surprising health finding in the history of medicine. Right up there with smoking causes lung damage and sitting in the sun for 8 hours is not a skincare routine. We have known stress is bad for us for decades. We have entire industries, meditation apps, wellness retreats, stress balls shaped like various soothing objects devoted to this single insight. And yet the actual mechanism, the specific biological story of what chronic stress is doing to your brain while you are busy being stressed about being stressed, is so much stranger and more alarming than the general warning label suggests that most people, if they genuinely understood it, would probably find it quite stressful, which is unfortunate timing. But here we are. The thing is, stress itself is not the villain. This is the part that tends to get lost in the wellness conversation. The stress response, that cascade of physiological changes triggered when your brain perceives a threat, is one of the most elegant and effective pieces of biological engineering in the vertebrate body. It evolved over hundreds of millions of years to solve a very specific and very urgent problem, which is what to do when something is trying to kill you right now. And for that problem, it is genuinely brilliant. Here is how it works. You are a zebra. You are standing on the savannah, doing zebra things, and a lion appears. Within milliseconds, your hypothalamus, a small but extremely bossy structure deep in the brain, has detected the threat and begun issuing emergency orders. The hypothalamus activates the sympathetic nervous system, which triggers the adrenal glands to dump epinephrine, better known as adrenaline, into the bloodstream. Heart rate surges, blood pressure climbs. Blood is redirected away from the digestive system and toward the large muscle groups. Glucose floods out of storage and into circulation, providing immediate fuel. Pupils dilate. Pain sensitivity drops. Every non-essential biological process, digestion, reproduction, immune function, long-term tissue repair is essentially put on hold. The entire body pivots within seconds. From its routine maintenance mode to a single overriding priority: survive the next 3 minutes. This is magnificent. If the lion is real and the next 3 minutes are the question, this response is everything you need. The zebra either escapes or it doesn't. The lion either eats or it doesn't. And the whole thing is resolved relatively quickly. Once the threat is gone, the stress response winds down. Cortisol levels drop. The parasympathetic nervous system reasserts itself. The body returns to its maintenance routines. Everything goes back to normal. Now, here is the problem. You are not a zebra, and your threats are not lions. Your threats are a mortgage that is slightly too large for your income, a job that feels perpetually insecure, a relationship that is slowly grinding you down, a commute that takes 45 minutes each way through traffic that seems to be getting worse every year, a news cycle that has apparently decided that the most efficient way to deliver information is to maintain your nervous system in a state of continuous low-grade alarm. These threats are not resolved in 3 minutes. They are not resolved in 3 days. Some of them are not resolved in 3 years. They are chronic, diffuse, psychosocial stressors, and your brain, which evolved on the savannah dealing with lions, does not have a separate biological response for this category of threat. It has one stress response, and it runs it on everything. So the same cascade that was designed to get you through 3 minutes of acute physical danger is being activated, repeatedly and chronically, by situations that last for months or years, and that cannot be resolved by running or fighting. The adrenaline surges. The cortisol floods in. The non-essential systems get suppressed. And then, crucially, the threat does not resolve, so the response does not wind down. It keeps running, and running, and running. This is where the brain damage begins. Cortisol is the primary glucocorticoid stress hormone, produced by the adrenal cortex under the direction of the hypothalamic pituitary adrenal axis. In the short term, cortisol is extremely useful. It mobilizes energy, sharpens certain kinds of alertness, and helps the body manage the demands of acute stress. But cortisol is also, over time and in excess, genuinely toxic to neurons. Specifically, it is toxic to the hippocampus. The hippocampus is a curved seahorse-shaped structure tucked into the medial temporal lobe, and it is central to memory formation, spatial navigation, and the regulation of the stress response itself. It is packed with glucocorticoid receptors more densely than almost any other brain region, which makes it exquisitely sensitive to cortisol. In the short term, this sensitivity is actually useful because the hippocampus uses cortisol as a signal to help encode stress-related memories. If something stressful happens and cortisol spikes, the hippocampus notes this and says, "Remember this, it was important." Survival-relevant memory formation. To make sense. But sustained chronically elevated cortisol does something very different to hippocampal neurons. It causes dendritic atrophy. The dendrites, which are the branching extensions that neurons use to receive input from other neurons, retract and shrink. Synaptic connections weaken. In severe and prolonged cases, hippocampal neurons die outright, through a process involving excitotoxicity essentially. The neurons are overstimulated to the point of exhaustion and death by glutamate, a neurotransmitter whose release is amplified by chronic glucocorticoid exposure. What this means, at the structural level, is that sustained chronic stress literally shrinks your hippocampus. Not metaphorically, not as a figure of speech. Measurably, physically smaller. This has been demonstrated in human subjects with remarkable consistency. People with major depressive disorder, which has extremely high overlap with chronic stress, show measurably reduced hippocampal volume compared to non-depressed controls, with the reduction correlating with the duration and severity of the depressive episodes. People with post-traumatic stress disorder show hippocampal atrophy that is detectable on MRI. Chronically stressed caregivers, people in high-pressure occupations, individuals living in conditions of persistent socioeconomic adversity, all show evidence of stress-related hippocampal compromise. And because the hippocampus is itself a major regulator of the HPA axis, it sends inhibitory signals that tell the stress response to wind down a damaged hippocampus becomes less effective at turning off the cortisol tap, which means more cortisol, which means more hippocampal damage, which means even less inhibition of the stress response. A beautiful, vicious, self-amplifying loop. And the hippocampus is not the only structure being remodeled by chronic stress. The amygdala, that ancient almond-shaped thread detection center buried in the temporal lobe, responds to chronic stress in the opposite direction. While the hippocampus shrinks, the amygdala expands. Chronic glucocorticoid exposure causes dendritic growth in amygdala neurons, increasing the complexity and connectivity of the threat detection circuitry. The amygdala becomes larger, more reactive, and more sensitive to potential threats. It fires more easily, more intensely, and takes longer to calm down after activation. So chronic stress simultaneously shrinks the brain region responsible for memory, learning, and stress regulation, and enlarges the brain region responsible for threat detection, fear, and emotional reactivity. The net result is a brain that is worse at learning, worse at remembering, worse at regulating its own stress response, and better at detecting threats and generating fear responses. If you were trying to deliberately engineer a brain optimized for suffering and dysfunction, this is roughly what you would design. And the prefrontal cortex, that crucial regulatory structure we keep returning to, also takes a hit. Chronic stress reduces synaptic density in the prefrontal cortex, particularly in the areas responsible for working memory, impulse control, and the top-down regulation of emotional responses from the amygdala. The very circuitry that allows you to think clearly under pressure, make good decisions, regulate your behavior, and tell your amygdala to calm down is structurally compromised by the sustained Stress that is supposedly the thing you are trying to think clearly about and manage. The cognitive tools most needed to cope with chronic stress are among the first casualties of chronic stress. This, at minimum, deserves some kind of award for biological irony. Now, let's bring this down to earth, because these mechanisms have real behavioral consequences that most people experience without understanding what is causing them. You have probably noticed, if you have ever been under sustained stress for an extended period, that your memory gets worse. You forget things you normally would not forget. You walk into rooms and cannot remember why. You read the same paragraph 4 times and retain nothing. This is not aging. This is not distraction. This is, at least in part, hippocampal compromise. The structure responsible for consolidating new memories is operating in a compromised environment, flooded with glucocorticoids that interfere with the cellular mechanisms of memory formation. You have probably also noticed that chronic stress makes you more irritable, more reactive, more prone to seeing threat and hostility in neutral situations. A colleague's tone of voice that you would normally not notice suddenly feels like a personal attack. A minor inconvenience produces a disproportionate emotional response. Traffic, which has always been annoying, becomes genuinely enraging. This is your amygdala, expanded and sensitized by chronic glucocorticoid exposure, running its threat detection algorithm on everything with the sensitivity cranked up past the point of usefulness. It is not a character flaw. It is a structural change in your threat detection hardware. And you have probably noticed that the decision-making that seemed reasonably manageable before the sustained stress somehow becomes overwhelming during it. Simple choices feel paralyzing. Long-term planning feels impossible. The future seems to compress into an immediate present where survival management is the only available cognitive mode. This is your prefrontal cortex running on compromised circuitry, struggling to perform the executive functions that require its full operational capacity. The cruel additional layer to all of this is what chronic stress does to neurogenesis, the production of new neurons. For a long time, the dogma in neuroscience was that adult brains did not produce new neurons. We were born with our complement, and that was it. We now know this is wrong, particularly in the hippocampus, where new neurons are continuously generated from stem cell populations throughout adult life and integrated into existing circuits. This adult hippocampal neurogenesis appears to play important roles in certain kinds of learning. Pattern separation, the ability to distinguish similar experiences as distinct memories, and mood regulation. Chronic stress, via glucocorticoids, suppresses hippocampal neurogenesis. It reduces the proliferation of new neurons and impairs the survival and integration of the ones that are produced. So chronic stress does not just damage existing hippocampal neurons. It also impairs the brain's ongoing capacity to replace and supplement them. Here is where the social dimension becomes impossible to ignore, because chronic stress is not randomly distributed across the population. It is heavily concentrated in people living under conditions of socioeconomic disadvantage, discrimination, housing insecurity, food insecurity, and exposure to violence. The neurobiology of stress does not care what is causing the activation of the HPA axis. It runs the same response whether the threat is a predator or an eviction notice. And the people most likely to be exposed to the kinds of sustained, uncontrollable, chronic psychosocial stressors that produce the neurological damage we have been describing are disproportionately the people with the least access to the conditions: stable housing, adequate nutrition, safety, predictability, social support that buffer the brain against that damage. This matters because we spend enormous energy as a society responding to the behavioral and cognitive consequences of stress-damaged brains. The impaired decision-making, the emotional dysregulation, the memory problems, the difficulty with future-oriented planning, as if those consequences were simply the product of bad choices or insufficient character. We do not ask what happened to produce this brain. We do not ask what the glucocorticoid levels looked like during the first decade of development, when hippocampal architecture was being laid down. We just observe the output and make our judgments about the person producing it. The relationship between poverty and brain development is one of the most well-documented and least acted upon findings in modern neuroscience. Children growing up in low-income environments show measurable differences in prefrontal cortex and hippocampal development compared to children in income environments. Differences that appear in early childhood and that are substantially mediated by the chronic stress burden associated with poverty. By the time these children reach school age, the neural architecture they are using to learn, regulate their behavior, and navigate social situations has already been shaped by years of glucocorticoid exposure that their more affluent peers simply did not experience. We then assess them using the same metrics, hold them to the same standards, and attribute the differences to individual variation in ability and effort. The neuroscience finds this approach about as sensible as measuring the running speed of people with broken legs and concluding that some people are just naturally slower. What actually reverses stress-induced hippocampal damage? This is where things get genuinely interesting. Exercise-sustained aerobic exercise is one of the most robust neurogenic stimuli identified in adult mammalian brains. It increases hippocampal neurogenesis, promotes the production of brain-derived neurotrophic factor which supports neuronal survival and growth, and has been shown to partially reverse stress-induced hippocampal atrophy in both animal models and human subjects. Antidepressants, particularly SSRI, also promote hippocampal neurogenesis, which has led some researchers to suggest that their therapeutic effect is mediated, at least in part through this mechanism, rather than simply through serotonin reuptake inhibition. Social support buffers the HPA axis response to stress, reducing the magnitude and duration of cortisol elevation. Environmental enrichment, novel experiences, cognitive challenge, social engagement promotes hippocampal plasticity and resilience. None of these interventions are primarily about willpower. They are about changing biological conditions. They work not because people decide to have healthier brains, but because specific inputs produce specific neurobiological responses that alter the structure and function of the tissue. Here is the realization worth sitting with. Every person you interact with today is carrying a brain that has been shaped at the structural level by their history of stress exposure. The amygdala sitting behind their eyes may be enlarged by years of threat. The hippocampus may be atrophied by sustained glucocorticoid exposure. The prefrontal connectivity may be reduced by chronic adversity. You cannot see this on their face. You cannot detect it from their behavior without knowing what you are looking at. But it is real, it is physical, and it is influencing everything they think, feel, remember, and decide. And your brain, shaped by your own history, is doing exactly the same thing. Right now, quietly, without asking your permission. The stress response evolved to save your life in 3 minutes. What it does to your brain over 3 decades is a different story entirely. And it is a story we have only just begun to read with sufficient honesty to understand what it is actually telling us.